just a quick bit of housekeeping for everybody. This meeting is being recorded, so just bear that in mind. If you don't want to be your information to be recorded, then please don't type in the chat or say anything. And also, if there's anything confidential, please do avoid putting anything confidential up, because it's this will be available to the general community. So anyway, welcome to the Midlands Mass Spectrometry Group seminar series. We've had a bit of a break over the summer, but this is our second seminar, and it's great to be back. And we've got two really excellent talks to kick off the new season. I just thought I'd say a couple of things about the Midlands Mass Spec Group for those of you who are not familiar with it. It's a collaborative partnership between the mass spectrometry groups and mass spec researchers across the Midlands and actually beyond the Midlands. So we include a few universities that lie outside the Midlands. And what it aims to do, what the Midlands Mass Spec Group aims to do, is to showcase the excellent mass spec facilities and research groups and groups using research we have across the Midlands, and to promote the use of mass spectrometry in research. Mass spectrometry has a huge role to play in research, and it's growing all the time, and we think it's really useful that we have a group that's able to promote that and to encourage people to get familiar with what mass spec can do and to use it in their research. So what Midlands Mass Spectrometry Group is able to do is it's also able to provide a really good first point of contact if you have any questions about mass spectrometry, or if you're interested in using it in your research, or even if you want to know what resources we have across the Midlands and what we're able to do. Or even if you just want to find out what mass spec could do to forward your research. We have the resources. We can point you to the groups around the Midlands, and the great thing that we have with the collaboration that we have across the Midlands is we're able to point you to the group that is best able to help you do the research that you want to do. So we don't sort of say, oh, we'll do it. We'll say, actually, the best groups in the Midlands are these groups, and you should go and talk to those people. And the other thing that we do, which we're very proud of, is that we support the excellent research technical staff that we have across the Midlands, without whom we would really not be able to provide the world's leading research and research support that the mass spectrometry groups are able to provide. So we have a very strong view on supporting research technical staff, and we work with things like talent in Midlands Innovation. So most importantly, the Midlands Mass Spectrometry Group is supported by Midlands Innovation. For those of you, I'm sure most of you will be familiar with it. It's a collaboration between 8 universities across the Midlands. That's Aston, Birmingham, Cranfield, Keele, Leicester, Loughborough, Nottingham and Warwick. And Midlands Innovation aims to drive cutting-edge research, innovation and skills development across the region. And we're really pleased that they support the Midlands Mass Spectrometry Group. And I'd like to thank the Midlands Innovation team for their help and support in both running the MMSG and in getting this seminar series together. So on to today's seminars. Today's seminars will follow the format that we used for the last one. We'll start with a talk from Dr. Jeb Bellamy Carter from the Leney labs in Birmingham, which is a little bit more focused on the technical side of things. And then we'll have a more outcomes-oriented talk from James Hughes from Helen Cooper's lab at the University of Birmingham, and that's the use in study of in biology and disease. There'll be time for questions at the end of each talk, and then at the end of the whole session, there'll be a bit of time for questions as well, if some haven't been answered, or if you have more general questions about mass spectrometry or about the Midlands Mass Spectrometry Group, or even about Midlands Innovation. If you have any questions, if you could type them, if you look at the bottom of the screen, you'll see there's a Q&A box down there. If you could type your questions in the Q&A, that would be great, because that helps us to keep them together. But if you forget and type them in the chat, we will pick them up from the chat as well. If you have anything really pressing that you want to say, so if you really feel you'd like to talk, then if you raise your hand, we can promote you to allow you to talk, to unmute your microphone if that's appropriate. So if you really want to do that, then please do let us know. So hopefully there'll be something for everybody in the set of talks we have this morning. So I won't take up any more of the time. I'll just get on and introduce our first speaker. So our first speaker is Jeb Bellamy Carter. Jed did his PhD and postdoc at the University of Nottingham before moving to Birmingham to work in Annika Leney's group in 2020. And Jed's going to update us on his work in a really challenging area of looking at protein-protein interactions. So over to you, Jed.
Thanks very much, Andy. So I hope I'm not going to disappoint people too much by focusing a little bit more on outcomes maybe than you may be expecting. Can I just check that you can see my slides okay?
Perfect.
Awesome. Okay, yeah, so as Andy's already introduced me, I don't think I need to say much more on that. The only thing I want to say really is that we're trying to do all this with native mass spectrometry in particular. So I think, there's my pointer. So I think I'll start quite simply with protein-protein interactions. So the majority of pharmaceutical targets are things that drugs tend to target in disease states are things like enzymes, receptors, and ion channels, singular proteins. But really, that's not how proteins function, and that's not really what they do in the cell. They're part of dynamic networks with a large number of other proteins. And it's the protein-protein interactions which typically mediate the majority of these. And to give you a feel for what that might look like, we're talking about some 700,000-ish protein-protein interactions in the human cell. And many diseases actually involve perturbed protein-protein interactions at PPIs, either those PPIs that have been disrupted because of disease mutations in the proteins themselves, or the opposite, forming of complexes which you don't really want to be there for normal functioning. And when it comes to trying to deal with these sorts of protein-protein interactions in disease states, that the majority of drugs that do exist for these are inhibitors, and that they fit into two basic classes, competitive and alloseric, where essentially a competitive inhibitor has a really strong affinity for one of the proteins. It will bind to it and it will outcompete the other protein, sort of as shown in this little diagram here. Alloseric inhibitors are very similar, but what they do is they modify the shape, the conformation of one of those proteins that makes it no longer able to interact. And the sorts of standard methods for probing these things are things like ELISA or SPR. I won't read through the whole list because I think you can do that yourselves. I think you'll be familiar with quite a few of them. But this is only really useful when you're looking at protein-protein interactions which have formed in disease states that you then want to disrupt. What about when a protein-protein interaction that's important for keeping the cell functioning has been disrupted itself and you want to promote that? So the solution to that really are stabilizers, or molecular glues, as we call them. And the way these tend to work, as you can guess from the diagram, is that a protein-protein interaction already happens. There's a certain affinity between the two proteins, but the stabilizer promotes that. It shifts the equilibrium over to this ternary complex. And a large number of these stabilizers can be sort of quite selective and self-limiting. And related to this is quite a few of the most interesting ones actually have really low affinity for either of the individual proteins. That's a very useful property because, again, it means that that's really specifically targeting this protein-protein interaction. You can have relatively high dosages. Unfortunately, it means that the sort of standard techniques don't work quite as well. So SPR, ITC, these can all be used to look at these things, but you need to use a range of concentrations. You have to do titrations, and to sort of to actually observe the ternary complex, often you need to be pushing towards saturation limits, which isn't necessarily reflective of the cell or what we're interested in. So what we're wanting to do really is come up with a rapid method for detecting novel PPI stabilizers. And these could come from really complex mixtures, extracts, or just a selection of different drugs. And one thing we're really keen on getting is stoichiometric information. Where are things binding and what sort of cooperativity might be going on? So this is where native mass spec comes in, and native mass spec really, it allows us to measure the mass. We're mass spectrometrists, that's what we do, of a protein or a protein complex. And when we've got sufficient mass resolution, we can measure multiple stoichiometries at the same time, as each one of them will hopefully have a different and unique mass, and we can pinpoint what that means in terms of the binding. And as a sort of small point, our techniques in native mass spectrometry tends to be more sensitive to small changes, and we can use relatively low amounts of sample compared to things like NMR, X-ray crystallography. So if I were to to do a little thought experiment. This is a mass spectrum in the mass domain rather than the M over Z domain of a protein-protein interaction. We've got two proteins, protein 1, protein 2, and this is what they might form. They might form this complex. Now, what happens if we add an inhibitor, the sort of the standard model? Well, this inhibitor binds to one of those proteins, it pulls that out essentially of this equilibrium, and then that releases more of protein 2 in this case. And this is the sort of shift you might expect in spectra. Alternatively, the stabilizers that we're so interested in, this is one where it's got very low affinity for either of the two individual proteins, but you can see that what we're trying to get is this formation of a ternary complex that's much greater in abundance than maybe you'd expect. So again, going back to the traditional techniques, the sort of things we're looking at is you measure the KD, the apparent KD, because they don't have stoichiometric information. They have to work on the sort of overall picture. You do it in the absence and presence of a stabilizer, and you essentially take the ratio of the two apparent KDs, and that gives you a cooperativity factor, alpha. It tells you basically where alpha is above 1, great, you've got cooperativity, you've got a stabilizer. When it's below 1, it's essentially an inhibitor. And as I've already kind of alluded to, this is the sort of thing we might expect with a mass spectrum if we were to be monitoring that sort of situation. So again, the ternary complex becoming more abundant is what we want. So just quickly, the way we're doing this, we take our receptor and a phosphopeptide mimic, in this case, I'll explain that in a second, of our partner proteins, that's what I'm going to call them for here. And then we do it with and without the stabilizer. And our actual instrumental setup is to do this by a nano-electrospray through a nanomate coupled to a HF, but really any suitable native mass spec setup would do. And we happen to deconvolute our data, and then we look at the relative abundances. And this helps to sort of simplify the spectrum and really get what's going on. And you'll see that soon. And we're doing this on a model protein, 1433, which I call a little bit of a swinger. And basically that's because it's a eukaryotic adapter protein, and it interacts with hundreds of proteins, all phosphorylated, or mostly phosphorylated, I should say. And this is a non-exhaustive list of some of them, and those highlighted are the ones we're currently interested in. And the reason we picked 1433 is it's very well known to have certain interactions that are stabilized or not stabilized by some small molecules. And one of these small molecules is called fusacokin A, or FCA. So the PPIs we're interested in are ER alpha, P53, and LRRK2. And I won't go into the biology, but we can talk about that later perhaps. But the one key thing to note is that 2 of these are known to be putatively stabilized by FCA, and one, LRRK2, is our negative control, it's not. So if we then do our native mass spectrum, just to convince you that the protein we're looking at is really doing the sort of things we might expect. This is just a typical native mass spectrum of 1433, showing that it's dimeric, it's native form. And then what we're doing, as I said, is that we deconvolute, we go to the mass domain. This makes interpreting the data a little bit simpler. So on to some actual stabilization data. Let's start with the negative control. This is our LRRK2. It has high binding affinity to the 1433, as we observe with our mass spectra. We can do another step of abstraction and convert deconvoluted data into these bar charts, which essentially show you the abundance of each of these stoichiometries. And when we add FCA, our putative stabilizer, not for LRRK2, we don't really see much of a change at all. We see some moderate binding. Now, what happens when we do this with ER alpha, putatively stabilized partner interaction? Well, this is the sort of thing that we see. ER alpha already has very high affinity for the 1433. It occupies both binding sites in a small amount. But when we add the stabilizer, everything shifts over. The equilibrium really pushes towards this ternary complex with the FCA very highly bound, the ER alpha present. And we can take this further to try and explore that. So we can start. So this is the same. These are the same charts from the previous slide. But what we can do is if we start, let's say we drop the concentration of ER alpha, but keep the other two things the same. What we then see happening is, as you'd expect, lower binding affinity of ER alpha, but still we get some stabilization from the FCA, but it's not as drastic a shift as we see when the ER alpha is more concentrated. We do the opposite experiment. We shove in a ton of FCA, the stabilizer, we really push it right over to this complete ternary complex of all the binding sites occupied by FCA and by ER alpha. The really interesting thing here, and we can talk about it a bit more later, is that when we observe the ER alpha bound to our 1433, once we've added FCA, we don't see it not also bound to FCA. What I mean by that is that the FCA seems to be being cooperatively bound to the 1433 at the same time that the ER alpha is being cooperatively bound. So they're working together to improve each other's affinity and binding to the 1433. But this experiment shows that we can relatively simply observe from these stoichiometries, we can tell exactly what's going on, how the occupancy of the different binding sites is affecting things. And as you kind of have alluded to, it's peptide concentration dependent. So this means that the two concentrations of the two proteins that we're interested in interacting do really matter in terms of what's going on. But in this case, it's telling us that there's a complex interaction with ER alpha and the FCA. So one of the things I said earlier was about complex mixtures. If we wanted to go from extracts or fragment libraries, we might want to screen multiple drugs simultaneously. So that's what we try to do. So this is our 7-drug cocktail containing a mixture of putative stabilizers. These are stabilizers that have been referenced in literature, and putative non-stabilizers, some other drugs we found in the lab. So FCA you've already encountered, and we're also interested in its analog FCJ. These are both extracted from the same source. We're also interested in PER1 and epistatin. Both of these have again shown some literature evidence of stabilization, but this is not from a stoichiometric point of view. This is from those sort of ITC style experiments. And this is a full mass spectrum that I'm showing rather than a deconvoluted one, because I wanted to sort of give a sense of the data we're looking at and the real wealth of information. We can pull out quite a lot of detail from our spectra. So that one thing to note is that we pull back out the FCA stabilization that we've had before. This is with ER alpha, but we're also observing binding and stabilization of the FCJ, its analog, although to a much lower extent. So this is an equimolar solution. So this is showing you really the extent of difference between the two. One of the really exciting things that you'll see just here and alluded to in this graph is that we actually observe a heterogeneous mixture. So we observe a complex which has both FCA and FCJ present. So we can really pull out two relatively strong stabilizers, observe them in the same spectrum and be able to separate them. Now, these other drugs that we observed here, we didn't really see much in the way of stabilization within this spectrum. And in fact, we just show that they have some binding affinity, but not really much beyond that. So I'll sort of conclude, and then we can talk a bit more about the data in the Q&A session a bit later. But we've got a new tool for studying these sort of PPI stabilizers. The little take home message of the biological context, at least, is that the FCA, we're compounding on literature data that shows that ER alpha binding is stabilized by FCA, and in fact, I think we've got some evidence that shows that the ER alpha stabilizes the FCA binding itself. It's a fully cooperative effect. Though I've not shown any data for it here, I do have some extra slides that I can talk about later, where we show that weak stabilization of P53, and again, pleasingly, our negative control LRRK2 isn't really stabilized at all, but we still observe the binding. And we can screen multiple stabilizers all at once, pick them out from a mixture, which is really nice in terms of high throughput screening. We can do this on sort of minute time scales with our setup. And this is all because native mass spec can provide a snapshot of the binding equilibrium of the stoichiometry that you don't get with other techniques. We can pinpoint in one spectrum, in one bit of analysis, occupancy of several different sites, and again, all of these stoichiometries, which give us a much better picture of what's going on. So I'd like to thank quite a lot of people. This is a project that's been collaborative between the University of Birmingham and the University of Leicester. So in particular, I would like to thank Annika and Manjari, who started some of the initial native mass spec work. She was a BMSS summer student Annika had, and then also Richard, who together with Annika put together the project and has a keen interest in protein-protein interactions. Jasvid for expressing the protein that we dealt with, and Marta and Yusuke for providing us with the drugs which are so important. I'd like to thank the rest of the Leney lab, the Cooper group, who are always ever helpful, and we have some really nice discussions, their responses. I'd also like to thank Todd and Jing Li for the advanced mass spectrometry facility at the University of Birmingham. They keep everything running, and literally without that, we wouldn't be able to do any of these experiments. And finally, I'd like to thank you all for listening. So the stuff I've talked about here and much more beyond is covered in our recent publication in Chemical Science. So if you want some more details that I don't manage to cover a little bit later on, please check this out or send me an email. Thanks for listening, and I'll take questions in the Q&A and also a little bit later. Thank you very much.
Brilliant, thank you, Jed. That's a really, really nice talk, both covering technology, so the use of these stabilizers, and some of the biological outcomes. So that's really, really fabulous start to today's meeting. So there is a question in the Q&A, so I think I'll start with that. It's from Todd. It says, do you have any confirmation that these complexes are formed in the condensed phase and not just during the electrospray process? So do they actually exist in cells?
Yeah, so that's an interesting question. So what we can say is that we observe it from our electrospray needles. Beyond that, we can't say exactly what's happening in the cells themselves, but we've confirmed some of the complexes that do form from sort of ITC style experiments. But I think the other thing to go back on is that LERC2, that negative control complex, we didn't observe that stabilization. So it's not just throwing in this drug forms all these non-specific complexes. It does seem to be something real going on there.
Great, thank you. I have a sort of question that I guess follows on a little bit from that. So the 1433 proteins are dimers. They have the two binding sites, and those two binding sites are independent of each other. So the 1433s are known to scaffold between many, many different proteins. So you've looked at a very limited set where yes. The data you showed was for one protein and one stabilizer. Have you started to get a little bit more complicated than that to see whether you can get selectivity in terms of not just one protein binding, but probably the more important interactions, which are the scaffolding ones, which bring different proteins together?
That's a really good question. So the answer is not yet, but that's something we're really interested in. So we have a few colleagues at the University of Birmingham that have inadvertently found 1433s stuck to their proteins, where they're looking at other things, they're pulling them out. So we're looking to work with them on some of those. And yeah, one simple experiment we can do going forward is chuck two of the peptides that we've got that are mimics for our proteins in and see what happens.
And so you could probably use a sort of fairly complex panel of phosphopeptides and look to see exactly what's happening in terms of there are some key scaffolding interactions that 1433 is known for that are important in disease. So you could get the phosphopeptides for those, and then look at how these stabilizers, because what would also be interesting is whether they not only stabilize a complex, but whether they prevent other complexes forming.
Yes, so there will be some essentially inhibitors, some modulators which prevent some of those protein-protein interactions from happening. As far as we know, FCA doesn't do that, but that's relying on ITC and other biochemical data. So yeah, it would be very nice to sort of flip the final experiment I showed, get, I don't know, 20, let's say 20 peptides, as long as they're mass resolvable, obviously, chuck them in with FCA and see what happens.
And then there aren't any other questions, but I have one more, which goes right back to what you said about mass spectrometry at the start. You said it was really good because it could work at low concentrations, which are probably closer to those that that occur in nature. Can you comment on the concentrations that you're working at and whether they are both realistic in terms of the proteins themselves and of the stabilizers that you could get into a biological system?
Yes, that's a good point. So what I'll do is I'll say that, so when they do the typical ITC style experiment or the SPR experiments, this is typically with protein on the order of 100 to 1 or 100 micromolar to 1 millimolar protein, and then they're throwing in 1 millimolar drug, which I would say is on the extreme end of quite a lot of these things. We're working much closer to sort of 5 micromolar protein. So the 1433 in this case, we were doing 5 micromolar, 25 micromolar peptide, and then 5 micromolar of the stabilizer. That was our sort of rough experiment. The main reason that the 1433 was lower is it's going to be interact with loads and loads of other things in the cell. So the realistic concentration that might be available for interaction with these proteins is lower, even though its actual concentration in the cell is probably on the order of a 50 to 100 micromolar.
Great. If we get time later, we might talk about sequestration and things like that, which are really interesting questions. James, did you want to ask a question?
I was thinking, so, you know, there are these diagnostics that exist now, so drugs bound to another prosthetic. And I was wondering, could you bind these stabilizing molecules to drugs with some sort of prosthetic, and would that increase their binding affinity to proteins, you know, in vitro in the far distant future? Well, that led to a second question, actually, probably a more important one. How close, you know, the drug binds to one part of a protein? Where does the stabilizer bind? Are they quite close, or are they quite disparate in terms of where they bind on the protein?
So your first question, I think the answer is yes. I think that's quite possible. The second one is probably easiest for me to just quickly show the slides again, if it's going to work. Is that going to work? Oh, typical.
You got to run the gauntlet now, yeah.
Yeah. So this is from a crystal structure of FCA with P53 in this case. So this is some already published data, and you can see that they occupy a similar binding pocket, and they're quite proximal to each other. Mm-hmm. So that's for this particular case. What you could do, as you were saying, sort of in terms of putting on prosthetic groups and modifying this, yeah, if you were to imagine that you pop something over here, you might then be able to promote a non-typical 1433 protein interaction, or to stabilize that. There's some work that Richard's been doing actually on modifying the 1433 itself, initially with this sort of warhead that attacks sort of a neighboring cysteine, and that he's got some data that's showing that actually further promotes stabilization from a protein perspective. So if you did an analogous thing and shoved that on the drug molecule, that might also promote things a bit.
Interesting.
Brilliant. Thank you very much, Jed. I think in order to get through the talks, we're going to move on to James now. So thank you for that, but hopefully there'll be a bit more time later on, and if people do think of any other questions as we go through, please do just put them in the Q&A or in the chat. Anyway, so without more ado, we'll move on to our second speaker today. So our second speaker is James Hughes. James did his undergraduate and PhD at the University of Birmingham and is now a research fellow in Professor Helen Cooper's group there. Just to sing his praises, James won a prize for his talk at the recent British Mass Spec Society meeting in Sheffield. So we're really pleased that we've managed to secure him. I'm sure he's in great demand after that. And he's going to go on today to tell us a little bit about the development of an improved mass spectrometry surface sampling technique that they've been developing in Helen's group, and how this can be used in the analysis of clinical samples. So over to you, James.
Thank you very much. I'll get this all set up. Everyone see my screen?
Yep, perfect.
Okay, I'll get the laser pointer. Yeah, thank you very much. So I'll talk to you today about our work on developing the Nano-DESI platform for the imaging of protein-protein and protein-ligand complexes directly from samples of non-alcoholic fatty liver disease. So to quickly introduce the presentation, I'll talk about non-alcoholic steatohepatitis, native proteomics, mass spectrometry imaging, and the platform that we're using, just to introduce those for people who aren't familiar with it. And then we'll move on to the results, and we'll take a look at some of the images that we've been able to collect using this technique, including images of protein complexes and protein ligand complexes. And then we'll talk about the MS2 experiments that we used to confirm that we were seeing, oh dear, it's got some sort of auto, you can never trust PowerPoint. But we'll move on to then talk about the experiments that we use to confirm that we do see these complexes. So non-alcoholic steatohepatitis is a disease associated with obesity that, I tell you what, before I get started, I'm sorry about this, but I just know that I'm going to be disturbed by the auto scrolling. I recorded this slide for BMSS a while ago. Does anyone know how you can turn that off? Otherwise it's going to be moving around all the time. Okay, that probably has done it. I apologize. Okay, hopefully that'll have fixed it. So non-alcoholic steatohepatitis is a disease of obesity. What happens is you get this accumulation of fat within the liver over a long time. Over the chronic time of disease, you develop this fibrosis. And there's currently no pharmaceutical treatment or easy way to stage the disease. So there's an unmet clinical need for both of those cases, for diagnosis and treatment. And so it's important to identify protein biomarkers for this disease to tackle that. So let's talk about native proteomics. So what is native proteomics? So we have already discussed it a little bit today, earlier with Jed's talk, but just to recap, so proteins in the body have these non-covalent interactions, or can have these non-covalent interactions with other proteins and with ligands. So what we have, for example, here is this cartoon of one protein in purple and another one in green, and they're bound together. And then they also have these non-covalent interactions with some ligands attached. And in imaging experiments that we've done in the past, using the least nanomate, for example, we use denaturing solvents to do our imaging experiments. And those have been very successful, but during denaturing, what occurs is the proteins unfold. And so you lose your protein-protein interactions, and you also lose your protein-ligand interactions. So you're losing biological information. And then just to talk a little bit more about the sort of impact that has in terms of mass spectrometry, for protein complexes, you have fewer charge states and fewer charges due to the less accessible protein. There's not as much accessible to the solvent. Whereas in denatured experiments, you'll have more charge states and higher charges because the protein is unfolded. And this has impact as well for the instrumentation. So if we look at these cartoon spectra of a denatured versus a native experiment, you'll see that for the denatured experiment, you would see these many charge states that are highly charged. So they tend to be in the 600 to 2000 M/Z range is where we used to do our experiments. Whereas in native experiments, you have fewer charges, fewer charge states, so a higher M/Z. And because you have fewer charges and fewer charge states, your ion current is concentrated under fewer peaks. So it can help with signal to noise as well to do experiments natively. So there's both biological and mass spectrometry advantages to doing native imaging. And for those who aren't familiar with native imaging, mass spectrometry imaging in general, mass spectrometry imaging attempts to map the location of a species throughout, for example, a tissue. So what you'll have is tissue on a slide, and you'll collect a mass spectrum at multiple defined points across that tissue, and then build up a heat map to relate the location of a species back onto that, XY space. So the nano-spray desorption electrospiralization platform that we're using here at Birmingham to do these experiments, it consists of a capillary through which solvent flows onto the surface of a piece of tissue. Proteins are absorbed from the tissue into the droplet, and then that droplet is carried away by an emitter to the orifice of the mass spectrometer. And you can see that here. So here is the capillary, which is delivering the solvent to the tissue surface. Then a emitter carries that solvent from the tissue surface to the orifice. And we raster this glass slide with tissue on it underneath this assembly here. And that's how we collect our images. So there are two samples that I'll be showing you today that we analyzed. One is a sample of non-alcoholic hepatitis, and the other is a sort of control sample, a normal liver. So you can see in the NASH, the non-alcoholic hepatitis sample, you have these regions of functional tissue called regenerative nodules surrounded by non-functional fibrotic tissue. And you can see in the normal donor liver, you have a much more homogeneous tissue. I mean, there are still, there are blood vessels and there's fine structure, but you can see hopefully the difference in these two images. So one of the advantages of using this Nano-DESI platform, although it's not unique to Nano-DESI, is that it is non-destructive in terms of its sampling. So this is a serial section of the analyzed tissue, and then this is the analyzed tissue. And this was stained post-analysis, and you can kind of make out the region that we analyze using the Nano-DESI platform. And if we zoom in, you can almost see the tracks where the solvent was moved along the tissue as it rastered. But more importantly, you can see that there are these intact structures still. It hasn't destroyed the tissue. So you can see this regenerative nodule surrounded by fibrotic tissue. So we used 200 millimolar ammonium acetate as our solvent system with 2 times the CMC of C8E4 as a detergent. So ammonium acetate is a solvent that is commonly employed in native sampling, and the C8E4 detergent helps both to extract extract proteins from the tissue and also to stabilize the protein complexes in the liquid and in the gas phase. We then use some source collision energy to help remove the detergent and the solvent from our proteins during the electrospray, and the image was collected with a lateral and vertical resolution of 200 micrometers. So this shows the averaged mass spectrum of the entire image, summarized all in just into one spectrum, as well as identifications for several proteins that we've been able to identify using MS2. And that includes these protein complexes here, this hemoglobin heterodimer, hemoglobin subunit beta and alpha, this calreticulin plus calcium complex, and this GSTA1 dimer. You can see that there's many more proteins in the spectrum waiting to be identified. There's a lot of information that we're able to extract from the tissue using this platform and solvent system. So let's start with some of the monomeric protein images that we collected from the normal tissue. So we have the fatty acid binding protein 1, profilin 1, Hep B1, NNMT, and PPIA proteins. So 4 of these you can see have this relatively homogeneous distribution throughout the tissue. And this is kind of what we anticipated for the normal tissue. We didn't expect to see many differences, but we were surprised with this NNMT protein, but this 30 kilodalton, almost 30 kilodalton protein. We were surprised to see this distribution throughout tissue. And when talking with our collaborators up at the Institute for Biomedical Research here at University of Birmingham in the liver labs, I was chatting to them about where is this being distributed throughout tissue, and it appears to be distributed in zone 1 of the portal region. So if you are familiar with the fine structure of the liver, it's basically a series of hexagons of this repeating portal structure, and zone 1 is the edges of those hexagons. So that's where it's being localized. So that was interesting. We didn't expect to see protein distributions necessarily in the normal liver. Of course, apart from, of course, hemoglobin signals, which you can see here. This is for hemoglobin heterodimer with signal localized to these blood vessels. So there's one here. You can see, I think that's the easiest one to pick out. The shape is very similar, as well as vessels here and sort of throughout the tissue where we're seeing this protein appear. And then we also have these two complexes, the RID-A homotrimer and the GSDA1 homodimer, this 40 and 50 kilodalton complexes respectively. These are homogeneously distributed throughout the tissue, and that's fine. This is a normal liver sample. But if we move on to the NASH sample with its much more heterogeneous sort of tissue structure, we can see that reflected in the images quite clearly. So again, we have the fatty acid binding protein wild type and variant. I'll talk about that in a second. The PPIA, PEDP1 profiling, and NNMT. So you can see in several of these images, there's this clear distribution that's localized to these functional regions rather than the non-functional tissue. And then you have the PPIA protein, which appears to be homogeneously distributed throughout the tissue. But coming back to this fatty acid binding protein, this wild type and this variant. So this variant protein is associated with worse outcomes in NASH and in non-alcoholic fatty liver disease. And you can read more about that here. When I was doing my PhD, we were using liquid extraction surface analysis to map the distribution of proteins in tissue. Although it's a very powerful technique and you can get a lot of information out of it, it does have quite poor resolution. And so we were never able to determine whether or not the distribution of these two proteins was different. We wondered if you would see the variant in some areas of the tissue and the wild type in others, and there would be a difference. But we can see with the improved resolution of the Nano-DESI platform that actually these two proteins do localize together. So that was kind of a nice answer to a question that I'd had for quite a long time about whether or not these proteins showed differential expression, relative to one another in diseased tissue. But moving on to the protein complexes, again, we have the hemoglobin alpha and beta heterodimer. This appears to be localized in these fibrotic regions, and during the generation of the fibrotic tissue, there is a lot of metabolism and vascularization as the tissue is laid down. So it's unsurprising to see these proteins localized to these regions. We then also have this calreticulin plus calcium. This again is a protein that when I saw the distribution for this protein and for the GSDA1, I spoke again to our colleagues at the Institute for Biomedical Research and asked them, as well as looking at the literature. And you see that both these proteins are again involved in tissue that is undergoing a lot of metabolism. And both of these proteins have also been shown in the literature to be differentially expressed in NASH or NAFLD tissue or NAFLD patients versus normal. So it's possible that these are indeed showing, they reflect the disease that is occurring in the tissue. You can see this is localized again in this fibrotic region, and the GSDA1 protein appears to be localized in this region up here rather than throughout all of the functional tissue. So it could be that the tissue here is, in the midst of fibrosis, it is in a worse condition than some of these down here. It's interesting. So how do we confirm that these protein complexes are complexes? We need to do MS2 experiments, both to identify them and to uncover their stoichiometry. So whilst you can do MS2 experiments using the Nano-DESI platform, it can be quite challenging due to signal. And also, if you are doing MS2 on heterogeneous tissue, you don't know scan by scan if your protein's going to be there. There wouldn't be much point, right? If the tissue is heterogeneous, you're not expecting your protein to always be present. So one thing we decided to do sometimes was decouple the MS2 from the imaging. And to do that, we used the Advion Traverse Nanomate robotic pipette system and used liquid extraction surface analysis to extract the proteins from the tissue, deposit them into a well. We used the same extraction solvent as we did for the imaging. And then we would take that extract and put it into a pulled emitter, which was gold coated, and perform Nano-ESI using that. And so we'll look at the calreticulin and the GSTA1 proteins and how they're identified. So first, calreticulin. So I isolated a protein peak, performed MS2, got a bunch of fragments. That's great. First thing you'll notice, the keen-eyed amongst you, is that all of these fragments occur in aspartic acid residue, which is a bit weird. You wouldn't usually expect to see that in top-down proteomics, but this has actually been noted before as something that happens when you are performing top-down fragmentation on native proteins. You tend to have this preferred fragmentation or this preferred location at aspartic acid residue. So that's some evidence that this protein is in its native-like state. And the other little thing that I noticed once I had identified this as calreticulin, went back to the MS1 data to have a look at the precursor peak, was I noticed that there was this difference between the theoretical mass for calreticulin and the mass that I had observed in the spectra. And this confused me for a moment until I started looking through the literature and realized that this calreticulin binds calcium. And if we look at the difference in mass between the calreticulin theoretical peak and the peak that we isolated for the fragmentation, the difference is a calcium ion. So that provided evidence then that we had this calcium binding. Then the GSTA1, which was a protein-protein complex. Once again, a peak was identified, isolated and fragmented, and we got the ID for the protein as GSTA1, and that's all typical. But evidence of it being a homodimer is when you have your isolated peak. If you apply a small amount of HCD energy, like 10% normalized collision energy, you'll not get fragments, but you'll get these dissociation products. So you have your dimer, you apply a small amount of energy, and the dimer is broken apart into its monomer subunits, and you'll see this pattern appear. And then you can use either deconvolution to figure out their intact mass, or if you're using high resolution mass spectrometry in your MS2, you can look at the charge states and work that out as well. And by finding the intact mass of the dissociation products, you can figure out the stoichiometry of your MS1 peak. And just to tease some data, so we want to continue to push the mass range. So the instrument that we use for these experiments is the Orbitrap Eclipse, and this can be operated in both a normal and a high pressure mode. And in this high pressure mode, we found that we bias the mass range to this higher M/Z region. And we again get this very rich MS1 spectra, and isolating peaks from this MS1 spectra and using proton transfer charge reduction to figure out the charge states, we're seeing these very large proteins. And I don't have a protein ID, but I can tell you that this 107.2 kilodalton species is a homotrimer. I've been able to get dissociation from it, but unfortunately no protein ID yet. So to conclude, we've been able to detect protein-protein and protein-metal complexes directly from tissue. And we want to continue to push the mass range by using the high pressure mode on the Eclipse. We're very confident we can do it. We just need the time to do the experiments. And so that's very exciting. So thank you all for tuning in today. I'd like to thank Professor Helen Cooper, Dr. Oliver Hale and Dr. Emma Sisley. Dr. Oliver Hale and Dr. Emma Sisley worked for me on this project. Dr. Oliver Hale has put the system together, and Emma Sisley's working with me as well on imaging various tissues. Both presented at BMSS, and if you are a BMSS member, you can find their talks. I'd like to thank Todd and Jing Li as well for keeping the lab running, as well as Professor Ian Stiles and Dr. Patricia Laylor, who are collaborators in other parts of the university, and also the Leney group as well. Shout out, they do great work and they're great people to share a lab with. So, and thank you to the funders. I'll take questions now.
Brilliant. Thank you very much, James. Again, a really, really nice balance between new technology development and some applications to biology, and some really fantastic data in there, and real challenge to think about what we can do with this. There's a question in the Q&A, so I think I'll start with that. It comes from Don Jones, and it says, what is the contribution of a hepatic blood to any of these analyses?
The contribution of hepatic blood, I'm not quite sure. Do you mean, I did my presentation just, okay. Did you mean blood that's left over after the sectioning of a tissue, or?
Yes.
Yes. So these tissue, we take the tissue blocks that we get from the IBR and section them. So there shouldn't be any surface blood left on the tissue. It should just be blood that's remaining in veins that's giving us that strong signal in the vessels. We actually see in other experiments I've done in the past, if you get very, very large veins, the blood is kind of washed out during perhaps the sectioning. So you don't get hemoglobin signals. So it's in the sort of the smaller and medium-sized vessels, you get these very strong signals for blood. I'm not sure if that answers your question, or if I've understood it correctly.
Well, I'm sure Don will get back to us if it didn't. There aren't any other questions in the Q&A just at the moment, but I'll ask a question if I can. It goes right back to the basics, actually. So you were looking at two different sample tissue types, the non-fatty disease and the normal, and those two tissues look quite different when you stain them. So how good is the technique at comparing two tissues? Do you get different ionization from the two tissues, differences just based upon the fact the tissues are different in their own right, or is the sampling pretty good at taking any tissue or working with any tissue?
So it's funny you should ask this question. So there was supposed to be a third tissue section that we included, that was fatty liver. And in fatty liver, if you remember my introductory slide, you get those swollen hepatocytes filled with fat. And when we were running the Nano-DESI probe over it, the fat leached out of those cells and actually formed a little droplet. And I have a video I took through a microscope of this droplet whizzing around on top of our solvent. It caused a lot of problems. With the NASH tissue and the healthy tissue, the sampling just, it works really well. We get good protein signal from it. The H&E staining differences might just be me adjusting the contrast in the images differently in the software. I know for the NASH and the normal, the sampling works perfectly well for both.
Great. So there's a question from Don, and then I think Neil Oldham would like to ask a question in person. So I'll leave that to the team to see if they can promote him. But I'll ask Don's question again. So would there be an advantage of using dopants to allow charge reduction actually on the tissue, or do you really need those multiply charged states?
So the multiply charged states do help us figure out the intact mass of the proteins from the MS1. We can also do that by using PTCR, but I don't know if we would feel there's any advantage to getting rid of all the charge states or pushing the charge states even lower. And if we push them lower, it might cause problems with the M/Z range. The Eclipse only goes up to M/Z 8000. So if we reduce them even further, we might actually push them off our radar, as it were, and we won't be able to detect them at all.
Great, thank you. So I think Neil can now unmute himself and ask his question.
Yeah, thank you. Sorry, I didn't realise I needed to be promoted to do that. Thanks, Andy. Great talk, James. I was interested in the fatty acid binding protein stuff. Do you see any fatty acids bound in your spectrum?
We would love to see fatty acids bound. We've never really been able to confirm if we do or not. We've seen other things bound. I don't know if I'm selling the keys to the kingdom, so I'll be careful now, but not fatty acids, unfortunately. But we have taken a look, and I did chase something a while ago, but it kind of led nowhere. But it would be really exciting if we could see fatty acids bound to them.
Yeah, I was particularly thinking whether the use of the detergent might displace them, and that might be an issue.
Yeah. So great. Thank you, Neil. So I just wanted to follow up on the detergent as well, and ask about the proteins that you see, because obviously you know, there are tens of thousands of proteins in the cell and you have a subset of them. So is that a selective sampling that you get from the tissue? So you just see what you see and it's a subset of the proteins? Is it to do with abundance and ionization? They're all there, but you just don't see some of them. And this is particularly for people who might be thinking about applying these sorts of techniques to their own research and whether they see their proteins or not.
Yeah, so this is something that we actually discuss quite a lot. How do we see more proteins? Because we know that they're extracted, and you know this from other people doing bottom-up work. You know, if you extract proteins and do bottom-up, Emma Sisley has a paper on that. You get, you know, 1,000 proteins. Why don't we see 1,000 proteins? And it must be a case of, you know, there's competition for ionization, there's competition in the AGC. So we use techniques like FAMES, for example, to try and increase the number of proteins that we see by providing some sort of online separation that's compatible with the instrument's duty cycle. And we've also been looking, for example, I showed you that PTCR spectra where we isolated a single peak, and actually when we performed PTCR, 2 species jumped out of it. So by using things like ion mobility or PTCR or other techniques, we're hoping that we can increase the number of proteins that we see. But yeah, there is definitely a challenge there. I guess there will always be a question of how much of a proteome will you really be able to see, you know.
It's time for one more question. I can't see anything in the chat at the moment.
Do you mind if I ask something?
No, of course, Jed, please do.
This is kind of a crazy question, really, but you just mentioned like that you can use PTCR and that's allowed you to see some other things. So what do you think the feasibility is if you kind of did a boxcar style experiment where you split the spectrum into, say, 20 bins and did PTCR on a big bin? Do you think that's feasible to then try and pull out loads of other proteins that might be there, or are you just losing too much?
So PTCR is pretty good in terms of not losing a lot, because when you start playing around with MS2 or MSN, you can start just losing ions, but PTCR is pretty good to us. It is an interesting idea, yeah, for sure, to bin the spectra, perform PTCR and see if you see other things. Yeah, definitely an interesting idea.
Brilliant, thank you. So we're nearly at time, so I'm going to have to wrap things up. I mean, I think we could probably go on discussing both of those talks for hours to come. So 2 really fantastic talks to start the next series of presentations. And so thank you both very much. I'm sure you will have excited a lot of people about the ways they could use these in their own research. And it's really great to see that mass spectrometry research in the Midlands is still very strong and still world leading. So thank you both for doing that. I'm sure that if people do have any burning questions that you'll be very happy that if they got in touch with you and asked those questions. I'm sure that you'd be happy to help them out if they were interested in implementing any of the things that you do. So brilliant set of talks. Thank you both ever so much for agreeing to do them and for presenting so well this morning. So thank you very much, and thank you to all of the audience as well for the great questions and for attending the meeting this morning. Thank you all.
Yeah, thanks everyone.
Thanks. Thank you.